This is Group 3, introducing Ladin, Ambrose, Arj, Emily, Eunice, Kushleen, and Demi. That's me. We are so excited to teach you about Apixaban. This is the checklist of topics we will cover today. With Eunice and Emily.
Remember, Emily, when I taught you about Apixaban?
What? Don't you mean when I taught you, Eunice?
Oh, let's not start this again.
Why? Are you scared?
No, why don't you tell me about the uses of Apixaban then?
So, Apixaban, brand name Eliquis, is an anticoagulant used to treat conditions caused by blood clots, including deep vein thrombosis, which is a blood clot forming in the deep veins of the body, usually in the legs, a pulmonary embolism, which is the blockage of an artery of the lung, prophylaxis of venous thromboembolism after hip or knee replacement, which is a deep vein thrombosis that dislodges and becomes a pulmonary embolism, or prophylaxis of stroke and systemic embolism in non-valvular atrial fibrillation, and at least one other risk factor. For example, previous stroke or TIA, symptomatic heart failure, diabetes mellitus, hypertension, and being over 75 years old. Apixaban has a risk of interactions with antiplatelet drugs such as ibuprofen and CYP450 inhibitors. Patients should also be monitored for signs of bleeding or anemia. Some of the common side effects associated with Apixaban listed in the BNF include anemia, hemorrhaging, nausea, and skin reactions. Those will make more sense when we discuss the structure-activity relationship, which we will go into later.
Wait, so do you know how the drug actually works?
So, Apixaban is an active inhibitor of factor 10A. The drug inhibits either the intrinsic or extrinsic pathway. The intrinsic pathway follows this flowchart after damage to the blood vessel when the platelets are in contact with the collagen in the blood vessel wall, started by factor 10 conversion. The extrinsic pathway follows this flowchart. It is initiated by damage to the tissue, but then factor 5 conversion begins the pathway. Fibrin fibres form long strands that bind the platelets together to form a dense web, which catches other blood cells and forms the clot. Thrombin-activated factor 13 enables the fibrin strands to adhere to each other.
So Apixaban targets factor 13, surely inhibits other proteins.
Actually, Apixaban is highly selective and reversible factor 10A inhibitor. It inhibits both free factor 10A and prothrombinase activity and clot-bound factor 10A activity. Apixaban works by inhibiting the conversion of prothrombin to thrombin, so the fibrin mesh cannot form. That's how the drug works.
Tell me something I don't already know. Like, how was it actually discovered?
Wasn't it developed, not discovered?
Oh, really? I didn't know that. Please do go on.
Well, for several years, the vitamin K antagonist warfarin was used as an oral anticoagulant to prevent or treat thrombosis. Regardless of its widespread use and low cost, there are several problems associated with warfarin as an anticoagulant.
Yeah, I've heard patients on warfarin required frequent blood tests for monitoring their blood glucose levels, and dose adjustments were often required. Warfarin also interacts with many medications, and patient overdose is difficult to treat. There are many deaths reported annually due to intracranial bleeding.
Oh wow, that's really heartbreaking. That's why Apixaban was developed, to address an unmet clinical need for a safe, effective oral anticoagulant drug that would not require routine monitoring for its appropriate use.
Oh yeah, the development of inhibitors of glycoprotein 2B and 3A, an integrin complex which aids in platelet activation, was heavily worked on by scientists. This led to their discovery of resemblances between the amino acid sequence of glycoprotein 2B3A and factor 10A. Once this was found, efforts were redirected to focus on factor 10A, which caused them to review archive molecules for better factor 10A efficacy. And this is what led to the identification of compound 1.
That's right. Once compound 1 was identified, techniques such as molecular modelling were used in the identification of a benzamidine-containing factor 10A inhibitor. This led to the development of compound 2, which displayed good potency. These are the functional groups that were changed. Compound 2 was significant in the discovery of compound 3 and compound 4, which were found due to reoptimization strategies. Compounds 2, 3 and 4 all demonstrated high antithrombotic activity in animals such as rabbits.
Oh yeah, the discovery of compound 4 led to the development of important compounds such as that of compound 5 and compound 6. Compound 6 is also known as Risaxaban. This was a huge breakthrough as it was the first small factor 10A inhibitor. Risaxaban acts directly on factor 10 in the coagulation cascade and does this without antithrombin as a mediator to provide sufficient factor 10A inhibition. However, risaxaban was not a sufficient warfarin alternative, as having 3 higher doses of risaxaban caused major bleeding to the patients in trials undergoing total knee replacement.
Yeah, that was so important. Without the development of risaxaban, the bicyclic pyrazole compound 7 would not have been discovered. This allowed for the addition of the functional groups that increase binding affinity to enzymes and increased effectiveness in the coagulation process. This change is seen in compound 8.
Did you know compound 8 showed good factor Xa affinity and anticoagulant properties in vitro? Compound 8 was also able to show high oral bioavailability and demonstrated impressive results in vivo antithrombotic animal models.
Finally, further development of compound 8 led to the discovery of compound 9. Compound 9 is known as Apixaban. It was found to be extremely potent and selective for factor Xa inhibition. Importantly, compound 9 showed an amazing pharmacokinetic profile in dogs.
Yeah, Apixaban was quickly progressed into clinical development as it showed an excellent preclinical profile.
That is so cool. I'm so glad patients with atrial fibrillation for whom warfarin therapy was unsuitable can now be prescribed Apixaban. The drug reduced the risk of embolic strokes without significantly increasing the risk of major bleeding or intracranial hemorrhage.
It truly is crazy that since 2007, development of Apixaban has been conducted in alliance with Pfizer. These novel oral anticoagulation agents allow physicians and patients to choose from drugs with profiles that differ from the traditional vitamin K antagonistic agent.
Isn't it? It's made such a huge difference, as now they can choose an agent based on its efficacy and safety profile to prevent mortality and serious morbidity in patients at risk of thromboembolic disease.
Thanks for reminding me of how Apixaban was developed. What else can you tell me?
Let's talk about how Apixaban was synthesized then.
Right, finally, I can teach you a few things. You see, the synthesis of Apixaban was derived from compound risaxaban. This synthesis contains 2 major steps. The first step, the readily available 4-methoxyaniline, or compound 1, undergoes diazotization. This is done by the conversion of the primary aromatic amine under sodium nitrate, concentrated HCl and 0 degrees Celsius, and is condensed in a solution with ethyl-2 chloroacetylacetate with sodium acetate to produce p-methoxyphenyl chlorohydrazone, which is compound 2. Compound 2 is reacted with compound 3 using excess triethylamine, which is a 3+2 cycloaddition. This is then treated with TFA dichloromethane to produce compound 4.
And step 2, I believe, is compound 4 is then treated with delta-valerolactam at 130 degrees Celsius for 24 hours under modified Ullmann conditions, using copper metal and thermal conditions. This produces the ester intermediate, which is compound 5. Compound 5, which is aminolysed with ammonia in ethylene glycol at 120 degrees Celsius for 4 hours to produce compound 6, which is Apixaban. Eunice, I just don't think you quite understand the structure-activity relationship of Apixaban.
Oh, I understand completely. You see, Apixaban is a factor Xa inhibitor. The S1 pocket of factor Xa is predominantly hydrophobic. Lined by amino acids such as serine and glycine, it contains an aspartic acid residue at the bottom end that serves as the recognition site for the basic side chain. For Apixaban, the amidine group is important for the aspartic acid interaction, forming a hydrogen bond such that the P1-aryl ether group in Apixaban fits into the S1 pocket of factor Xa. P2 in Apixaban binds to the S2 site, which is a small, shallow surface depression lined by tyrosine. The amide group of Apixaban can form a hydrogen bond with the phenolic group of the tyrosine. This is also common with the amide group of rivaroxaban and edoxaban, other drugs in the same class. P3 occupies the S3 site, which is flat and exposed to solvents. This is why it is vital for this part of the compound to be completely planar.
I hate to break it to you, but you missed a key binding site.
No, I didn't. Which one could I possibly miss?
Well, the entrance of the S4 pocket has an aromatic box generated by the arrangement of 3 aromatic amino acids, tryptophan, tyrosine, and phenylalanine. This forms pi-pi stacking interactions with the benzene groups as well as the gamma-lactam group of P4 in Apixaban. This is a key group of all the drugs in this class, where edoxaban uses the aromaticity of a pyrrole and rivaroxaban uses the benzene. The cationic hole in S4 is blocked by the Apixaban molecule at P4. The gamma-lactam group orientates the molecule to fit into the active site of factor Xa correctly. The carboxymido moiety binds directly to the active site and is important as it increases increases the drug potency and pharmacokinetics. Hydrolysis by non-selective enzymes is prevented by the bicyclic pyrazole. This is a characteristic not present in rivaroxaban, which gives Apixaban a much longer half-life.
Going back to the side effects, some of the common side effects listed in the BNF include an increased risk of bleeding, nausea, some skin reactions, and anemia. Now, the bleeding is due to the inhibition of factor 10A, which prevents blood clots causing excess bleeding in wounds and increased bruising. There is also an increased risk of anemia due to an increase in platelet count, and this in turn leads to less blood cell cultures due to the bruising and bleeding out of vital blood components.
Oh, touche.
At least we can both agree the development of Apixaban plays a vital role in the category of anticoagulants.
Yeah, I just told you that.
Fine, I give up. I guess we both know about Apixaban.
So we covered the structure of Apixaban, the biological basis of the disease, how Apixaban was developed and synthesized, the mechanism of action including the structure-activity relationship, and Apixaban's main side effects on and off target.
Yeah, we covered a lot, but now I have actual work to do, so goodbye.